every day, late at night, not okay. All I want, and I pray, all I need are some better days. Fuck me, I'm looking in the mirror, so foggy, but I've never seen clearer. I don't really think anyone can save me, and honestly I'm not really sure I want saving. I like to be my own worst enemy. There's no risk if you don't try at anything, so I'ma just keep buying everything. See you in the next life, have to be a better me. I don't think that my head's on straight, gotta flip it and grip it and go and get an x-ray. What's wrong with me? I just feel weight pushing on my chest and it squeezes till I suffocate. Better change my mindset, meditate. It's pretty cool that I'm alive and have better days. I could walk, see, hear, I should celebrate. Think I could change my mind, maybe elevate. Living life, every day, late at night, not okay. All I want, and I pray, all I need are some better days. Yeah, all I need are some better days. Cause all I want, and I pray, I believe in the better days.
Hello, good morning, and welcome to today's lecture by BB Science and Medical Media. Today, we are going to look at a very important topic. We've all heard about DNA, and as soon as a lot of people hear about DNA, what comes to mind is DNA paternity testing to determine the paternity or the father of a child. So this morning, we are going to look at how this paternity test is done, how it is carried out in a laboratory. So this will be our presentation outline. We'll have an overview of what DNA is. We'll also look at the principles of inheritance, how you inherit DNA. And we'll look at what is a DNA paternity test at all. You've been hearing about DNA paternity test. What is it? And what parties are involved in a DNA paternity test? Or what individual do we take samples from when we are going to do a DNA paternity test? And we look at what samples we can take for a DNA paternity test. And we will look at the processes or the various procedures involved in DNA paternity test. That is the various procedures that take place at the laboratory. And the procedures are DNA extraction, DNA quantification, DNA amplification, electrophoresis, DNA profile visualization, then the profile analysis and comparisons. And we also have a look at the statistical analysis or calculations involved in DNA paternity testing. Then we will have a look at how a DNA paternity test report is written. So, overview of DNA. What is DNA? So it's a DNA or deoxyribonucleic acid. It's the hereditary material in humans and also almost all other organisms. And nearly every cell in a person's body has the same DNA. So DNA stands for deoxyribonucleic acid, and it's an organic chemical of complex molecular structure that is found in all prokaryotic and eukaryotic cells, and also in many viruses. And most DNA is located in the nucleus of a cell, and in this case it is called nuclear DNA. But then a small amount of DNA can also be found in an organelle called mitochondria. where in this case it is called mitochondrial DNA. And we all know mitochondria is the powerhouse of a cell. Mitochondria are structures within cells that convert the energy from food into a form that cells can use. And DNA is made up of two length strands that wind around each other to resemble a twisted ladder. And this shape is what we call a double helix. So DNA codes for genetic information for the transmission of inherited traits. And within a cell, this DNA that we are talking about is organized into dense protein DNA complexes, which we call chromosomes. Now in eukaryotes or higher organisms, the chromosomes are located in the nucleus. And as we've already said, some DNA can also be found in an organelle called mitochondria. And also in plants, some DNA can be found in chloroplasts. Now in prokaryotes, and we all know prokaryotes, they do not have a membrane-bound nucleus. So in prokaryote, the DNA is found as a single circular chromosome in the cytoplasm of the cell. And there's one important property of DNA, and this property is that DNA can replicate or make copies of itself. So each strand of DNA in the double helix that we talk about can serve as a pattern for duplicating the sequence of bases. This is critical when cells divide because each new cell needs to have an exact copy of the DNA present in the old cell. Principles of inheritance. What is inheritance? In biology, inheritance can be defined as the process of how a child, we are using a child because in this case, we are interested in DNA paternity test. But then generally we will say how an organism receives genetic information from the parent. So the whole process of heredity is dependent upon inheritance. And it is the reason that the offsprings are similar to the parents. This simply means that due to inheritance, the members of the same family possess similar characteristics. It was only during the mid-19th century that people started to understand inheritance in a proper way. And this understanding of inheritance was made possible by a scientist called Gregor Mendel. So he conducted a series of experiments and brought out some laws. So Gregor Mendel formulated certain laws to understand inheritance, which today these laws are called Mendel's laws of inheritance. So between 1856 to 1863, Mendel conducted the hybridization experiments on the garden peas, that the pea plants. During that period, he chose some distinct characteristics of the peas and conducted some cross-pollination or artificial pollination on the pea lines that showed stable trait inheritance and underwent continuous self-pollination. Such pea lines are called true breeding pea lines. Mendel selected a pea plant for his experiments for the following reasons. 1. The pea plant can be easily grown and maintained. Also, they are naturally self-pollinating, but can also be cross-pollinated. A pea plant is an annual plant, therefore, many generations can be studied within a short period of time. And also, the pea plant has several contrasting characters. So, after Mendel's experiments, he brought out these conclusions, or these conclusions were gotten from Mendel's series of experiments. So the genetic makeup of the plant is known as the genotype. So if we bring it to a paternity test, between the genetic makeup of an individual is known as the genotype. On the contrary, the physical appearance of the plant, in this case the physical appearance of a person, is known as the phenotype, how the person appears physically. Now, genes are transferred from parents to the offspring in pairs, known as alleles. So, parents to offspring in pairs, meaning the offspring inherits one allele from each parent. During gametogenesis, when the chromosomes are half, there is a 50% chance of one of the two alleles to fuse with the allele of the gamete of the other parent. So 50% chance. So it means each parent donates 50% of an allele of a child. When the alleles are the same, that's when each parent donates the same allele to a child. They are known as homozygous alleles. And when the alleles are different, that's when each parent gives different alleles to a child. These alleles are known as heterozygous alleles. So, what is a DNA paternity test at all? You've been hearing about DNA paternity test. What is DNA paternity test? Now, as we've already talked about, DNA is a genetic material that one inherits from his or her mother and father, that's both parents. In this case, the DNA is called a nuclear DNA, found in the nucleus of a cell. And a DNA paternity test is a way of determining whether a man is another person's biological father. That is a way of determining the father of a child. So a DNA paternity test uses what we call DNA, which is usually taken from a cheek swab, or what we call a buccal swab inside the cheeks or inside the mouth, to determine whether a man is the child's biological father. And DNA paternity tests are extremely accurate. So who are the parties involved in DNA paternity tests? If we are going to do DNA paternity test, which group of people are involved, or are presented for sampling? The first person is the alleged father, the man in question. Then the second person is the child. Then the laboratory analyst can decide to include the mother. What it means is that DNA paternity test can be done without involving the mother. But then you can also choose to involve the mother. So you can choose to sample the mother as well. Sampling for DNA paternity test. How do we sample these parties that we just talked about for DNA paternity test? What sample do we take from them? The first sample is a buccal swab. And if you look at the picture here, this is how a buccal swab is taken. So the person opens his or her mouth wide. And then the laboratory analyst insert what we call a cotton swab, which is sterile or devoid of any contaminant, into the person's mouth and then rub inside the person's cheeks. All what this laboratory analyst is trying to do is to get the cells of the person. As we all know, DNA is found in a cell. So rubbing this cotton swab into the person's cheeks, you get the person's cells, and then you can extract DNA from these cells. Now, you can also use venous blood or a blood drawn from the person's veins in EDTA tubes. EDTA is an anticoagulant. So once you draw the blood from the person's vein, you put in this EDTA tube so that the blood will not clot. So this is a venous blood in EDTA tube. And the third option is blood on FTA cards. Now, an FTA card is a chemically treated filter paper which is designed for the collection, preservation, and shipment of biological samples for subsequent DNA analysis. Collection, shipment, and storage of biological samples when using FTA cards is very convenient. FTA chemistry has been optimized to lyse cells or break cells and denature or destroy proteins upon application of the biological sample onto the card to protect nucleic acids from nucleases. Nucleases are enzymes that can degrade or destroy nucleic acids. Also, to protect nucleic acids from oxidation and UV damage. So after drying, the released nucleic acids remain immobilized in the fibers of the matrix and are preserved for transport and later processing. The most important obstacle of processing the samples from the card matrix is the localized trapping of the genomic material after application of the sample. Moreover, long-term storage of the sample might hinder the release of the entrapped DNA from the card matrix for molecular diagnosis. So this is an FTA card. So these two places have been used. Blood has been applied to these spots. So this is an FTA card with blood sample on it. So these are the various options available when we are going to have a DNA paternity test. Now, after a sample has been taken, or after taking a sample from the parties involved, let's say the alleged father and the child, or the alleged father, child, and the mother. Now the samples enter the laboratory. So, in the next step, we are going to talk about how the samples are processed at the laboratory. After taking these samples, the first step is what we call DNA extraction or DNA isolation. It simply means taking the DNA out of the sample that we've already taken. DNA is found in the cell. And if you take a buccal swab or a blood sample, these samples contain cells. So DNA extraction means breaking these cells and taking the DNA out of the cell. So DNA extraction is a method to purify DNA by using physical and or chemical methods. DNA is separated from cell membranes, proteins, and other cellular components. The use of DNA isolation technique should lead to efficient extraction with good quantity and quality of DNA, which is pure and devoid of contaminants. It means the technique that the laboratory or the analyst is going to use to extract DNA should be highly efficient so that it will lead to good quantity in terms of concentration and also quality of the DNA, so that the DNA will be pure and devoid of contaminants. Now we are interested in DNA. So if any other thing gets into your final elute, then it means your DNA has been contaminated. So anything aside DNA is a form of contamination. Let's say proteins, RNA, they should all be degraded because we don't need them. What we are interested in is DNA. So in your laboratory, if you are also interested in proteins, it means when you are extracting your protein, DNA also becomes a contaminant. That's how it works. So what are some of the extraction kits or extraction methods that we can use in getting DNA out of a cell? In fact, there are several methods. There are a lot of them. One is the backbind blood and tissue DNA ADQ96 kit, which is manufactured by a company called Omega in the US of A. We also have the KLIAMP DNA Investigator Kit, manufactured by a company called QIAGEN. QIAGEN is a German company. And we also have the Profiler Forensic DNA Extraction Kit, manufactured by a company called Applied Biosystems. So this is the KI-AMP DNA Investigator Kit, which is manufactured by company. The company QIAGEN. You can see QIAGEN written on it. And these are the various components of the kit. These components have their roles to play in the DNA extraction process. In our next presentations, we'll go deeper into DNA extraction and we'll look at the importance of all these reagents, or to a layman, what is called chemical, in the DNA extraction process. The second kit is what we call the Magmine DNA kit, which I told you is manufactured by a company called Omega. You can see the Omega written on it. So it also has its components in this box. And this is the Profiler Forensic DNA Extraction Kit, manufactured by a company called Applied Biosystems, written over here. You can see Applied Biosystems. And these are the various components that this box comes with. And they all have their roles to play. As I said in our next presentation on DNA extraction, I'm going to go deeper into the importance of all these components and their role in the DNA extraction process. Now, after getting our DNA, or let's say we've broken these cells and we now have our pure DNA, what is the next step that goes on in the laboratory when we are doing DNA paternity test? Now, the next step is what we call DNA quantification. And DNA quantification simply means knowing the amount of DNA that we have. We've broken the cells and we now have our DNA. But then what is the amount of DNA that we have? What is the concentration of DNA that we have? We have to determine that. We call it paternity test or any other DNA test. we work within limits. Too little or too much DNA can interfere with the DNA process. So we need an amount of DNA. So we have to quantify to know if the DNA is more, then it means we have to dilute it to the concentration that we need. If it is less too, then it means we have to work around it to generate a DNA profile. So DNA quantification is a very good step. And with even modern methods, when you quantify, you're able to know whether what you extracted, the DNA you have, contains contaminants like the proteins and any other contaminant, or even if they contain inhibitors, what we call PCR inhibitors. Or even if it's not a paternity test and it's any other test, when you quantify using some methods, you are able to know whether the sample has even been mixed, or whether the sample or the DNA you have is coming from a male and a female source. That's a mixed sample. So DNA quantification is very important before you proceed to the next steps. So DNA quantification is commonly performed to determine the average concentration of DNA in a sample prior to proceeding with the various downstream experiments. Sample purity is also an important consideration to accurately calculate the amount of DNA in a sample. So choosing the right technology for your samples, workflow and throughput results in accurate DNA quantification and can save significant time and money by helping to prevent downstream experimental failures. So reliable measurement of DNA concentration is important for many applications, not just in DNA paternity test. Of course, DNA paternity test is also part of molecular biology, but then any other molecular biology application. And what are some of the methods that you can use or is being used to quantify or know the amount of DNA that we have? The first method is spectrophotometry. Now, spectrophotometry is a method to measure how much a chemical substance absorbs light by measuring the intensity of light as a beam of light passes through sample solution. The basic principle is that each compound absorbs or transmits light over a certain range of wavelengths. This measurement can also be used to measure the amount of a known chemical substance. The second method is fluorometry. Fluorometry is defined as the measurement of emitted fluorescent light. Fluorometric analysis is a widely used method of quantitative analysis in the chemical and biological sciences. It is a very accurate and sensitive technique. Fluorometry is superior to spectrophotometry, the one we just talked about, in terms of its sensitivity and specificity. The third method, which is currently being used by most laboratories in DNA paternity test to measure or quantify DNA, is what we call real-time PCR. And real-time PCR is a technique of choice for nucleic acid quantification. It's also called quantitative PCR. PCR means polymerase chain reaction. And there was a presentation on that, so you can check it out from my channel, DNA amplification or polymerase chain reaction. So real-time PCR is called a quantitative PCR. This means you are able to monitor what is happening in real time. That is why it is called real-time PCR. So it is a method by which the amount of the PCR uploaded can be determined, as I said, in real time. So over here is the spectrophotometer. So this is where you place the sample. You lift this side up, you place your DNA on it, you put this thing back, then it will read your concentration for you. Over here is the real-time PCR machine. This is a 7500 or 7500 real-time PCR machine manufactured by the company Applied Biosystems. So it is connected to a computer here with the software on it. So you open the software, you select the kit you are going to use. You can use a lot of kits. This is one of the kits. Quantifier Trio DNA quantification kits, manufactured by the same company, Applied Biosystems. So you use this kit, then you prepare your sample. Over here, you open here, you place your sample and you close it. And then it will read. The software will work on your sample for you. And using this particular quantifier Trio kit will let you know whether your DNA is degraded or not. It comes with something called degradation index. It will also tell you whether your DNA has even been contaminated. Let's say if you have a male DNA, it will tell you whether it's been contaminated with female DNA. If you have female DNA, it can tell you whether it's been contaminated with male DNA. To tell you, as I said earlier on, whether your DNA is degraded or not. It will tell you whether it contains PCR inhibitors. PCR inhibitors can interfere with your downstream applications. So this kit is very important, and real-time PCR quantification of DNA is very, very, very accurate and important. It can detect the little amount of DNA you have, unlike the spectrophotometer. And this is the fluorometry that we talk about. This is called a qubit fluorometer, manufactured by the company Invitrogen. So these are the things that with spectrophotometer, real-time PCR with its kit, then the qubit fluorometer. So now we are done with DNA extraction. We have our DNA. We also done a DNA quantification. We know the amount of DNA we have. The next step in the laboratory is DNA amplification. Amplification means just getting a lot of copies of the DNA. With this DNA paternity test, we are not going to amplify the entire DNA sequence. We have specific sequences on the DNA that we are going to target or amplify, because that is where it differs from human to human. There are a lot of places in our body that are the same, and there are some few places in our body that differs from individual to individual. So when we are doing this DNA paternity test, those places are where we target, or where our case are going to target, because research has been done and it's been proven that these places in our body differs from individual to individual. I mean, the sequences differs from individual to individual. So you can have some of these kits targeting, let's say, 11 sequences. You can have other kits manufacturers having their kits targeting, let's say, 16 sequences. You can have other kits targeting 24 loci. So the term is loci, that's the plural form, and individual that is called locus. So you can have other case targeting 32 loci. So there are various kits manufactured by various companies for this particular work, at the DNA amplification. And in the modern world we live today, the polymerase chain reaction, or the PCR, is very important for this particular work. It is the PCR that is being used to amplify or make copies of these target sequences that we are interested in. So DNA amplification is a powerful technique that has had an immense impact on scientific research, not just DNA analysis alone, not just DNA paternity tests alone, but then on several scientific research in the past 2 decades or even more. How PCR is still the most popular method, alternative methods of DNA amplification are constantly being developed. And this PCR is sometimes called molecular photocopying. The PCR is a fast and inexpensive technique used to amplify or copy small segments of DNA. Because significant amounts of a sample of DNA are necessary for molecular and genetic analysis, studies of isolated pieces of DNA are nearly impossible without PCR amplification. And in PCR, you can either amplify a single target sequence, or you can do what we call multiplexing. That is bringing a lot of target sequences together and amplify them at a go in just a single reaction. That is what we call multiplexing. So in recent years, there are commercial kits for this purpose. You don't have to buy individual reagents or components for your PCR. So when you come to DNA paternity test, we have a lot of commercial kits available that you can use. Some examples are the Identifier Plus kit, which targets 16 sequences or loci. We also have the Global Filer kit, which amplifies 24 loci. These two kits are manufactured by a company called Applied Biosystems. We also have the PowerPlex fusion system, which also amplifies 24 target sequences or 24 loci, which is manufactured by the company called Promega. So this is one of the most commonest PCR machines, or what we call a thermocycler. This is called a Gene Amp PCR system 9700-9700, manufactured by the company Applied Biosystems. So you use this case in preparing your samples. You load them here to the PCR machine, you cover it with this. Then the PCR comes with a lot of parameters, a lot of processes. When you watch our video on PCR, you understand this better. It goes through an entire process, then the target sequences are amplified. So this is the global filer kit that we talk about. It is the Identifier Plus kit, and this is the PowerPlex fusion system or kit from Promega. So now we've extracted our DNA, we've quantified it, we've amplified our interested sequences. The next step is electrophoresis, or separating these our sequences that we have amplified. So when you come to the laboratory workflow, this is almost the final stage of the DNA paternity test process. So electrophoresis is a laboratory technique used to separate either DNA, RNA, or protein molecules based on their size and electrical charge. An electric current is used to move the molecules through a gel or other matrix. So pores in the gel or matrix work like a sieve. So in this case, it is called a molecular sieve, allowing smaller molecules to move faster than larger molecules. So if you put molecules of different sizes into a sieve, you'll find out that the smaller molecules will move faster in the sieve compared to the larger molecule. That is how electrophoresis works. And the two most commonest methods of electrophoresis are the gel electrophoresis and also what we call capillary electrophoresis. In modern labs, this method is used, the capillary electrophoresis. So, what is a gel electrophoresis? Gel electrophoresis is a laboratory method used to separate mixtures of DNA, RNA, or proteins according to molecular size. In gel electrophoresis, the molecules to be separated are pushed by an electrical field through a gel that contains small pores. Now, what about capillary electrophoresis? Capillary electrophoresis is an analytical technique that separates ions based on their electrophoretic mobility with the use of an applied voltage. The electrophoretic mobility is dependent upon the charge of the molecule, the viscosity, and the atom's radius. So these are some of the machines that is used in electrophoresis. So these are all capillary electrophoresis machines. This is the 3500 XL genetic analyzer manufactured by Applied Biosystems. So you prepare your samples using your PCR product or your amplified DNA. You put in this machine and then electrophoresis is done for you. This is another version, a lower version of this machine from the same company. And this is also another version of the same genetic analyzer from the same Applied Biosystems company. And this is how a gel electrophoresis is also done. This is a gel tank. So you prepare a gel, normally an agarose gel using agar. Then you put in this tank. You fill it with the required buffer. You have wells in the gel. So you place your DNA in the wells. An electrical current is applied. Then you see the DNA running down here in the various wells. So they form lanes in here. So let's say if you have 5 or 8 wells over here, they all run down this place. So this is a gel tank. So you've separated your amplified DNA or you've done electrophoresis. How do you visualize it? How do you see it in real life? With capillary electrophoresis, you can visualize your DNA profile with softwares. So a typical software is a GMapper IDS software. And also in gel electrophoresis, you can visualize it under UV. So you have what you call a UV trans illuminator. So this is the GeneMapper IDS software. You open the software, then you log in your details. This a higher version of this software. You log in your details, then you can use the software. And this is the UV trans illuminator. So you can see the bands here from the PCR that we did. These are the DNA bands. So lane 1, lane 2, lane 3, lane 4, lane 5. And these are the various bands are being visualized in this UV trans illuminator. So after using the GeneMapper IDS software, that's after doing the capillary electrophoresis and do your analysis with the GeneMapper IDS software, this is how a typical DNA profile looks like. So those of you who have not seen how a DNA profile looks like, this is how it looks like in its raw form. Normally, when you have a DNA paternity test report or a DNA report, they or they bring out what is on this particular profile onto or into a table form. So most of you who are not scientists, or most of you who have not seen a DNA profile before, you don't get to know how the natural DNA profile looks like. So this is how it looks like. It comes in this form. So these are the segments or where we target in our body that I talk about, or the loci that I talk about. So this is one locus, another locus, another locus, another locus, another locus, another locus, another one, another locus, another locus. So these are the various segments in our body that research has been done, and we've seen that they differ from individual to individual. So during DNA paternity test, these are the various places or segments in our body where we target, amplify, and then look at the DNA profile. So this DNA profile is coming from a female. Why? Because if you look at the sex-determined locus, or what we call the amelogenin, you can see only X. We know females have XX chromosome and males have XY. So the X here, in reality, it is XX, but then the software will give it as X. So once you see it, it means your profile is coming from a female. If it's coming from a male, you'll see XY at the amelogenin. So when you have a raw DNA profile like this, the only thing that you can say about this DNA profile is that it's coming from a female looking at the X. Without this particular locus, there was no way this DNA profile would have been meaningless. But if you look at this, then you know it's coming from a female. And also we have another locus called Y-indel. This analysis is done, or this particular locus is found on the Y chromosome. So we have nothing here because females do not have the Y chromosome. So when you look at the Y-indel, there was no number. That's to the layman. These numbers mean something, but to the layman, you can call it numbers. So there was no number here, or the numbers are what we call the allele. So there was no allele here because the female doesn't have a Y chromosome, and this particular locus is found on the Y chromosome. Once again, you can see DYS391. This is also another locus on the Y chromosome. That is how come you don't see any allele or any number here, because this is a female DNA profile, or the sample that we generated DNA profile, this DNA profile from, is a female sample. So you can have 17 here. If you remember in our presentation, we talked about homozygote, that's Mendel's experiment, the conclusion from Mendel's experiment. if you inherit one allele, or to a layman, let me use the word numbers. If you inherit one number, as in this case 17, from both parents, then it means you are homozygous for this particular locus. So you can see 17 here. It means a person inherited 17 from the mother, at the same time inherited the same 17 from her father. That is how come you only see 17 here. So this 17 means 17, 17. It means 17 from both parents. Just that the software recorded it as 1. So this is the meaning of the homozygote, inheriting the same thing from both parents. Let's look at this locus, VWA locus. We can see 15, 17. It means the person inherited one allele or one of these numbers from both parents. It's either she inherited 15 from the mother and 17 from the mother and from the father, or she inherited 15 from the father and 17 from the mother. So this is what is called a heterozygous. So the person is heterozygous for this particular locus, 15, 17. So you can conclude from this place, it means this place too, the person is homozygous, because the person inherited the same number, that is 9, from both parents. Once again, another homozygous peak over here. Then when you come to this locus, the person is heterozygous. The person inherited different numbers from both parents. So this is how a typical DNA profile looks like, that from capillary electrophoresis. Now, once again, as soon as you see this profile, let's go to the amelogenin locus. You can see SY. So this is a profile coming from a male. And once again, if you look at the locuses on the Y chromosome, because this is coming from a male, you can see an allele at the Y indel. You can also see another allele at the DYS391 locus, but we didn't see these alleles in the female sample or the female profile, because females do not have the Y chromosome. But then we are seeing alleles from these loci, 2 loci, this one and this one. You can see 2 here, and you can also see 10 here, because this profile came from a male, and males have the Y chromosome. So this is a profile coming from a male. Now, these are what we call the bands. That is DNA profile or electrophoresis using the gel electrophoresis. This is how it looks like. So these are the wells that I talk about. 1, 2, 3, 4, 5. So you put your PCR product in each well. So if from this picture, the PCR profile from the mother was put in well 1, PCR profile from the child was put in well 2, and then 3 men were tested. It means it is alleged that any of these 3 men is a potential father for this child. So the 3 men were tested. So man 1's PCR product was put in lane 3 or well 3. Man's 2 PCR product was put in lane 4 or well 4. And the third man's PCR product was put in the 5th lane or the last lane. Now this is how the comparisons were done. You can see this red rectangle over here. This is the mother, and the child has the same band at the same level. It means the mother gave this band to the child. Now, if you look at the child's next band, this is the next band coming from the child, also corresponding to a band from the mother. It means, once again, the mother gave this particular band to the child. If you look at the third band coming from the child, it doesn't correspond to any of the mother's band. It means the band from this child came from the father. So automatically, if any of these men is supposed to be the father of this particular child, then that man should have this band, because the man donated this band from the child. If you look here, the mother do not have this band. Now, man 2 has this band corresponding to the child, and man 3 also has this band. So if you look at this man, it means man 1 is out of the question now. We are left with man 2 and man 3 as the potential fathers of this child. Let's continue. Now, if you look at the last band from the child and compare it to man 2 and man 3, now man 2 has a band corresponding to the child. Over here, the mother do not have this band, so this band should be coming from the father. And man 2 once again has the same band. Man 3 do not have this band. So we've settled it now. So we can conclude that man 2 is the father of this child. That is how the the gel electrophoresis also works. They come in bands like this. Thank you. So we've already done the profile analysis and comparison for the gel electrophoresis. If you go back, that is what we just look like. This is how the profile analysis and comparison is now. So now let's go on to the profile analysis and comparison for the capillary electrophoresis. So how is it done? Child's profile against alleged father's profile. So you compare the two DNA profiles. We have the child's profile, then you compare it to the father's or the alleged father's or the supposed father's profile. So what do? You look for the common alleles at each locus. So you compare the two profiles and then you look for the common alleles or the alleles, or in simple words, as we saw from the profile, the numbers that appear at each locus in the man's profile and the father's profile. Now, where the mother was tested to, as I said, you can do DNA test without the father. You can also include the mother. So the second one is child's profile versus mother's profile versus the alleged father's profile. You check for something called obligate paternal alleles. Now, the obligate paternal alleles are the numbers that I show you. So these are alleles of each test locus that the alleged father must have if he is the father of the child. So the obligate paternal allele is deduced by comparing the mother and child's profile. So if the child has an allele at a test locus that is not present in the mother's profile, that allele will be an obligate paternal allele. Why? This is because 50% of DNA is passed on by a child's mother and 50% is also passed on from the father. And therefore, since it didn't come from the known mother, the allele must have been passed on to the child by the unknown father or the supposed father. So that is what we call obligate paternal allele. So when you are testing three parties, you first look for obligate paternal allele. So you take what the mother contributed to the child, then what is remaining in the child profile should definitely come from the father. So that is the obligate paternal allele. That's what you look for in the father's profile. So let's use this as examples. So this is a DNA paternity test conducted between a child and an allele father. This test was done without the mother. So first of all, let's go back. As I said, if you have just a child and an allele father, you look for common alleles at each locus. So you go to each locus and look for common alleles. So this is how you see a DNA report. Now the raw profile that I showed you earlier has been brought here in a table form. So this is the child's profile, this is the allele father's profile. So the two numbers means heterozygous. Where you see a single number, it means homozygous. We've already talked about, or we've already explained that. So you already know this, heterozygous and homozygous. Thank you. So this is the STL loci. These are the various segments or the various loci in our body where we test. So this kit is actually is the global phyla kit from Applied Biosystems. It has 24 loci. So this is a DNA profile generated using the global phyla kit. So let's look at the first locus, child has 15, 17 as the alleles. The father has 15, 16. So do they share common alleles? Let's see. 15, 15 is also here. Yes, the common allele is present. We go on to the next locus. The child has 13, 20. The father has 16. They don't share any common allele, so the common allele is absent. The next locus. The child has 9, 11. The alleged father has 11, 13. 11 here, 11 also here. It means the common allele is present. The next locus, 7, 11. 7, 12. They have 7 in common. Common allele present. The next one, 8, 9. 9, 11, they share 9 in common. Common allele is present. The next one, or the next locus, the child has 14. The father has no 14. It means the common allele is present. They don't share any common allele. The next one, 28, 30. 28, 30. They share the common allele. It's either the 28, 28 or the 30, 30 because we didn't test the mother. The next locus, 16, 19. 15, 21. There's no common allele here. Let's come here. 11, 14, 12. No common allele. The next one, 7, 9.3. 9. No common allele. So this is how a DNA profile comparison is done. When you test only the child and the alleged father, you look for the common alleles. Now, if the father or the supposed father or the alleged father is indeed the father of the child that we are testing, we should be seeing common alleles at all the loci that we test. If only one, after looking at these 24 loci and only one doesn't match, we can attribute to something called mutation. But then, if we find this absent more than twice, that's 2 times or more, then you should know that the alleged father is not the actual or the biological father of the child. So over here we can see how many absent? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Definitely it means this father can never be the biological father of this child. Let's go on to the second one. So once again, this is a test between a child and an alleged father. And over here we can see there is a common allele at every locus that was tested. So common allele present, 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 present throughout. It definitely means that this child is the child of the alleged father because they share common allele at all the loci that we tested. So this is a positive DNA test. And over here you can see some paternity index. We'll have a presentation on this so that you understand more into this. A paternity index is one of the calculations that is done to determine whether a supposed father is the real father of a child. So the paternity index has been calculated here. From this table, the previous table, if you look at the paternity index column, you can see 0, 0 here. Where there was no shared or common allele, the paternity index over there is 0. We didn't calculate for these ones, though they were present, but then it doesn't make sense because at the end of the day, we will multiply everything and anything times 0 will be 0. So that's why we didn't calculate for these areas because all these places were 0. So the final answer, that's the combined paternity test, after multiplying all of them, will be 0. That's why there was no calculation for these places. So now let's move on to 3 parties paternity test, where we tested the child, the mother, and an alleged father. So now let's look for the obligate paternal alleles. As we already talked about, obligate paternal allele simply means you first take the allele donated by the mother out of the child's alleles. then the remaining allele from the child should definitely come from the father. That's the obligate paternal allele. So let's look at the first locus. The child has 17. It means 17 from both parents, homozygous. Let's look at the mother. The mother has 16, 17. So the mother donated 17. So the other 17 should come from the father. The father has 17. So the obligate paternal allele, bill 17, is present. Let's go on to the second one. The child has 16, 17. The mother has 16, 20. Definitely this 16 should come from the mother because the mother has no 17. And the remaining 17, which is now called the obligate paternal allele, should be coming from the father if the father is indeed the biological father of this child. Let's look at the father's proof. The father has 17 here. So over here, the obligate paternal allele is also present. The third locus. The child has 9, 12. The mother has 8, 12. It means this 12 should definitely be coming from the mother. Because normally we assume that mothers are the real mothers of children. So 12 should be coming from the mother. Remaining 9, it means the 9 being the obligate paternal allele should be coming from the father. The father has 9 here. So the obligate paternal allele is present. Let's go on to this place. 8, 12. The mother is 8, 12. So definitely the mother donated one of these. Now the father has 10, 12. There was 12 here. So it means the 8 is coming from the mother and the 12 is coming from the father. The child has 9, 11 here. The mother has 9. It means automatically the mother contributed the 9 and the 11 should be coming from the father. The father has 11 here. So obligate paternal allele is present. So you can do same for all this. So obligate paternal allele was present at every locus tested. Then you can know that the alleged father is the real or the biological father of the child in question here. And the paternity index has been calculated here. Another one, three-party DNA paternity test. The child, the mother, the alleged father. Over here, the child is 16. The mother is 16. It means the mother contributed 16, and 16 should be coming from the father as well. But the alleged father doesn't have 16. So the obligate paternal allele being 16 is absent. The second one, 15, 18. The mother has 18. It means 18 coming from the mother. 15 is the obligate paternal allele and it should automatically come from the father. The father has 15. Obligate paternal allele present. Same for here. Let's come to CSF1PO locus. The child has 11, 12. The mother has 8, 11. It means 11 should be coming from the mother. Remaining 12, which is the obligate paternal allele. If you look at the father's allele, the father has 8, 9. The father has no 12, so obligate paternal allele is absent. So you can do same for everything here. So how many absent? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 out of 24. Then you should know that the alleged father is not the biological father of the child. If you look at the paternity indexes, once you have absent, it means paternity index after you will definitely be 0. So 0, 0, 0, 0. That is why we didn't do calculation for this place, because at the end of the day, you are going to multiply everything, and any number times 0 is 0. So there are also a lot of statistics involved in DNA testing. But then before we go on, please don't forget to subscribe to this channel for me. Subscribe to this channel. Don't forget. Also like this video, comment on it. Any question that you have, leave it in the comment section and I'll try my best to answer you. Also, any comment that you have in terms of having, maybe you're also in this field and you have comments, you can leave it there. Also share this video for it to go viral so that others will also see this video. So let's take some few seconds to subscribe. I'm waiting for you to subscribe so that we can move on. It's very important for me, please. You should all subscribe to this channel. Thank you. So I hope you've all done the subscription. So in our statistical calculations, we use something called allele frequencies. There will also be a video on that so that you understand it better. And we use something called paternity index. We've already seen that. And then we combine all the paternity indexes, or we multiply them. Then we get what we call a combined paternity index. After that, we calculate what we call a probability of paternity. That is what you've been hearing about. That, oh, this person was 99.99% for paternity, this person was 0%. That is what we call probability of paternity. So these are the things involved in our statistical calculations. And this is very important because it is going to tell the entire world the probability of a person being the father of a child compared to other untested men. So let me give you a brief about what an allele frequency is about. So the allele frequency represents the incidence of a gene variant in a population. Now, alleles are variant forms of a gene that are located at the same position or genetic locus. So in this case, the numbers that we see, those are the alleles, the numbers that we saw earlier on. Or that are located on the same position or genetic locus on a chromosome. So an allele frequency is calculated by dividing the number of times the allele of interest is observed in a population by the total number of copies of all the alleles at that particular genetic locus in the population. So this is a calculation. Allele frequencies can be represented as a decimal, a percentage, or a fraction. In a population, allele frequencies are a reflection of genetic diversity. We also have paternity index. And the paternity index, and we normally represent this by the letters PI, that's paternity index. This is the genetic odds in favor of an alleged father being the biological father of the child. A paternity index is calculated for each genetic locus. As you saw earlier on, the calculation was done for each locus and represent the likelihood that the alleged father contributed the paternal alleles versus an untested, unrelated random man in the population. A combined paternity index, CPI, which is calculated by multiplying the individual paternity indexes together, summarizes the overall strength of the genetic evidence. For example, if the combined paternity index is 100,000, it means that the alleged father is 100,000 times more likely to be the biological father of the child than an untested, unrelated, random man in the population. In other words, the chance that he has matched the child by pure coincidence would be just 1 in 100,000. If the alleged father does not possess the paternal alleles of the child at 3 or more loci, then the paternity index at each non-matching locus is 0. So definitely the combined paternity index will be 0. And the alleged father is excluded as the biological father of the child. So report writing. After going through all this, how do you write a DNA paternity results? Now the conclusion of the report has two things. We have the inclusion results, that is the alleged father being the real or the biological father of the child. And we also have the exclusion results, that is the father or the alleged father ruled out as the biological father of the child. So probability of paternity we normally get 99% and above. Then when the father is not the biological father, definitely you are going to get 0% because some of the paternity indexes will be 0 and we will have a combined paternity index of 0. So the results indicate that the alleged father cannot be excluded. That is the inclusion results. It means the alleged father cannot be excluded as the biological father of the child. and this result is based on statistics calculated for the comparison. Even so, it is not correct to say the father is the biological father of the child, because once you say that, it means there was 100% probability, but normally because of the statistics involved, you get 99% and above. So there was still a small chance, a very slim chance that another man might be the father. But this is science or statistics. So once you don't get 100% probability, you cannot say you are the father or the other man. So what we normally do is that we write it in such a way that you avoid that 100% probability. So therefore, the results are reported as the alleged father cannot be excluded as the biological father of the child. In simple words, it means the alleged father is the father of the child. Now, the second result, that's the exclusion results. The results indicate that the alleged father is not the biological father of the child. We are using is not because over here we have a probability of paternity to be 0%. So there was no chance that the father is the biological father. 0% means 0%. So over here we write a definite statement that the results indicate that the alleged father is not the biological father of the child. This conclusion is based on the absence of 2 or more alleles. You go to some labs, they will use 3 or more alleles. Others will use 2 or more alleles. Normally it is 2 or more alleles found in the child that were not donated by the father. So as I said in our presentation, if you have one locus not matching, we can attribute it to something called mutation. But then 2 and above, then we can conclude that the alleged father is not the father of the child. So these are the conclusions that are drawn or that are written in a report. So in the report, you have the samples taken, the report to state the sample that were taken, whether blood or buccal swabs. Then the report will give the table form, as we saw there, comprising the STR loci, the allele called the paternity index. Then the report will also indicate the calculation, that the probability of paternity. Then the conclusion, whether inclusion or exclusion. That is how a DNA paternity test report is written. But then the most important part is the conclusion part, whether inclusion results or exclusion results. So now, when the results is an inclusion or inclusive one, we say the alleged father shares common allele with the child at all loci tested. Then I'm using an example of a paternity test that I've already done. In this one, the combined paternity index was 1, 2, 1, 3, one whole lot of number, a very huge number. We use a probability of 0.5. Probability, the probability we assume before even testing. Before even testing, there was a 50% chance that the alleged father might not be the father. There was another 50% chance that the alleged father might be the father. So we include what we call a probability of 0.5 in our statistical calculation. Now, I had a probability of paternity for this particular one to be 99.9999%. So the conclusion is that I drew was that the alleged father cannot be excluded as the biological father of the child, because cannot be excluded because I didn't get 100%. I've already explained that. That is the language we use in DNA paternity test. Now for exclusion results, we say the alleged father does not share common allele with the child at some loci tested. That is when you have some of the loci. If you have some of them, you look at them and there was no common allele shared by the father and the child. Then you can say the alleged father does not share common allele with the child at some loci tested. You can decide to mention those loci involved. Definitely the combined paternity index will be 0. probability 0.5, probability of paternity 0%. Now you can draw your conclusion that the alleged father is excluded because this is 0%. So over here you are making a strong statement. The alleged father is excluded as the biological father of the child. Ladies and gentlemen, finally we've come to the conclusion of our presentation or lecture. So this is how a DNA paternity test that we've all been hearing about is done in a laboratory. You go through the process of sample collection, DNA extraction, DNA quantification, amplification of our target sequences of interest, electrophoresis. You visualize the profiles, you do your statistical calculation, profile comparison, you write your report, you draw your conclusions. This is how the DNA test or DNA paternity test that we've all been hearing about is carried out in a laboratory. Thank you for your audience. I really appreciate you for watching and listening to this presentation or lecture. Once again, don't forget to subscribe to this channel. That's very important for me. Help me build my channel. Encourage me by subscribing. Comment and share this video. Also like. Thank you very much and have a wonderful day. Thank you.